I don't want, it's too much time to do that now. All right. The outline is going to look like this. This replication. It's awfully dark. You see. Yeah. I'll paint some more. But it's replication of DNA. And you've got to replicate DNA for a cell to divide. I mean, my cells have, every single cell's got 46 chromosomes. Well, to make a brand new cell, he could have 46 too. So before a cell divides, he got double the chromosome number from 46 to 92. And then half of them go this new cell, half of that new cell, and those cells gone, of course, because I got cut in half. And my new cells, they have 46. Well, there is a way that the cell replicates. And that's what we're going to talk about. And I'm going to get into it right now.
It's not here yet.
And do a little talking. And then when you download it from Angel and you start filling things in, you go, oh yeah, I recall him saying that.
You gonna put it after class?
Huh?
Put it Angel after class?
I'm going to put it on between now and my last class tonight. I'll put it on when I get home today.
Okay.
I'll probably put it on, I can put it right here. I'm sitting here and put it on Angel real fast. All right. This is my calendar page, and it's the coolest thing around. This is the DNA right here. I need two of these things. So what happens? This is where you find the hydrogen bonds. It unzips. These bases match. See, there's a T right there. You see that T right there? Mm-hmm. T right there. So these move up and plug in on both sides, and they glue together on the sides. When you get done, you have two brand new DNA. Half of it was the original one, half of it is new. So if this is A side, there's A and there's B, right? Well, the original A is right here. There's your new B. There's your original B. There's your new A. And there's a name for that. I'll tell you later on what it's called. But that's how DNA makes a copy of itself. All right. You can jot the words down and just listen, because I'm going to give you both to later on anyway. Replication, duplication, to me means the same thing. You've got to make an exact copy of all your DNA so that when you divide, that new cell you produce will have the same thing you have. Now, mitosis produces twins. There won't be any change in cells. I mean, if our reproductive was based on mitosis, we would all look identical. But we do meiosis, and meiosis is a shuffling of genes. So you don't look like your sister or your brother, your mom or your daddy. But if we use mitosis to make reproduction, we'd all look the same, like twins would. See, twins do mitosis at first. And that's why when they get born and they're identical, they look alike. Now, you got two strands. There's your two strands. There's one strand, I call it A. There's one strand, I call it B. Okay? So you got two strands, and this one is the complement of that one. If I say up, he says down. If I say black, he says white. I mean, they're the very opposite of each other. And that's how this whole thing works. These little T's are called bases. So that's a base, that's a base, that's a base, that's a base. They're all bases. And this base joins this base with hydrogen bonds. And those bonds are easily broken. So we can easily unzip and then zip right back up again. I do this all the time. If you get cut and you got to heal by making more cells, then the cell does not hurt, unzips itself, makes a copy of itself, and that goes into a brand new cell, and eventually you heal because the cells will make copies of themselves. You gotta remember that the red in this picture was the original, and the blue what you put on now. As far as the body concerned, these two are identical twins. They're identical twins because basically this one would be the same as that one. So when you get done, both of these are just like that one was. That's called replication. Okay? I'll tell you how the partner work in a minute. Now, it's much easier taught than it is shown because this is a complicated process. And I'll tell you the easy way and the hard way. Well, I'll tell you the easy way and the real way. Because the real way is the hard way. Okay. There's a pattern. You only have 4 bases. You only have 4 bases in all the DNA. You only have 4 bases. And they're called complementary. And like I showed you, with those red and blue, when it's over, the 2 brand new ones look just like the one. Now, see where it says semi-conservative? What's semi mean? True or almost?
Yeah.
It means almost. And here's what I mean by that. How much of this one was the original? Half. How about this one? Half. That's why they're called semi-conservative. Because only half of it was the real one. The other half is the thing we added. So that's not totally conservative, it's semi-conservative. Okay? That's what we're talking about. But when you're over now, those 2 brands, I know they're red and blue, but each one looks just like that one that started. The same codes, and I'll show you how the codes work in a minute. You may already know about A, B, and all that A's, T's, and C's, G's. Now, complementary.
What?
It's right here. It's right here. There are only 4 bases in DNA. Let me back a minute. You see these T's right here? They can be an A, or a T, or a C, or a G. That's it. I should put one more there, I guess. But these bases, even these bases, could be an A for adenine. Or it could be a T for thymine. Or a C for, what's it called?
Cytosine.
Cytosine. Or a G for
guanine.
guanine. You remember how they pair up?
Mm.
A always hooks to who?
T.
T. C always hooks to
G.
Now, we learned that because when they began breaking these things down, they found like, man, there's a ton of A and a ton of T, and a little bit of C and a little bit of G. Well, if the A is hooking to G, it'd be the same amount, wouldn't it? So if I say let's say I got 100 bases. Let's say I have 30 and I have 30 A's and 30 T's. How many is that in all? 30 and 30 is
60.
Now of my 100, how many also must be the C's and G's? 20 and 20. Now, if I know that the A is 30, then, and I know that the T is 20, then I know the other 2 must have been. So we know that way that the A's are always hooking the T's, because you wouldn't have 30 A's and 20 T's, would you? They wouldn't match up. So if you have 30 A's, you're always going to have 30 T's. All right? So that's what we're talking about. And most of you, those are the words. Now these are just for DNA because RNA has one oddball. And when I list them, it's how they pair. I just do that naturally because, and what I do, I'm saying A at T. The word at is my remembering device. A at T, AT. If I remember that, then I know that G and C is go together. Okay? Now, and like y'all told me, A always hook to T or vice versa. If you see a T, what goes with it is A. If you see an A, what goes with it
G.
T. If you see a G, what goes with it?
C.
C. Or if you see a C, what goes with it?
G.
But that's called the complementary base pairing. And this is my clue right here. That word right there is my A and my T coming together. That's how I remember it. Okay? Now, let's look at this. Let's look at this piece of DNA. Notice always 3 bonds between these two. Always 2 bonds between those two. That's why they can't get messed up. It's like you cannot plug a 3-pronger into a regular 2-prong socket. It won't fit. And because these have the bond, this is always a triple bond between the C's and G's. These are always, if this was 3-2, that might be a problem. They could get all around. But by being 3 versus 2, that guarantees. Now, they ain't gonna mix up. They can't hook up wrong. Now, in the case of this little circle and that like home plate baseball and this thing here, these 3, that's a nucleotide. These 3 things are a nucleotide. Or these 3 things are nucleotide. Or these 3 things. Notice how they point up this side and point down that side? They don't all point up. Did you catch that? On this side, now these are sugars, by the way. They're always pointing up. On this side, they're always pointing down.
Complementary.
And if you look at the 5 and the 3, you point toward the 5' end. You always point to the 5' end. So the flat part is on the 3' end. The flat part, and that makes a difference how they come back together. But that, I'll tell you later about that though. But notice that when you put it together, both sides ain't pointing up. And some folks wouldn't caught that. One side points up, and one side points down. It always goes 3 to 5. 3 to 5.
Are you going to tell us about what the 3 and 5 mean?
I am,
Okay.
but not yet. I'm going to this been.
Okay.
All right, now. This is how it actually works. And I showed you, you see this thing right here? Where there's a carbon on all these corners, there's a carbon. There's carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Okay? If you look carefully, the phosphorus is hooked to the carbon 5, not the carbon 3. That's how I know that these are always, that's the unit. Because the carbon is hooked, this is number 5 right here. These hooked together. That's how I know that this is a unit, because there's your carbon 5 right there. So carbon 5, I'm pointing right there in a way. Actually, carbon 5 is up here. Carbon 5 is sticking off of the base. Here's your base. And carbon 5 is right there. I say base, I mean like a baseball home plate. Okay? That's not, the base is over here really. Okay. Now, so the 5 means that the phosphorus that connects these 2 together is hooked to this one. Now, you see that O right there? And that one? This H will hook to the next one in line. It's gonna be hydrogen bond, so that when you end up, they alternate phosphorus, sugar, phosphorus, sugar, phosphorus, sugar, phosphorus, sugar, and the bases come off of the number 1. The number 1. And so when you build this thing, it runs this way, up one side, 3 going to 5. If you flip it over, it runs, it's like 2 one-way streets. One goes this way, and one goes this way. They're not two-way on one highway. Okay? So all these numbers identifying are the carbons that are important, and basically it's the number 5 carbon that hooks onto. And then this line there, I should have one there. I don't know why I forgot to put it there. It should be one there too. There should be a bond right there. It's not by itself. I just forgot to put a bond in there. Okay. Now this is the next one. I'm just telling you what this does. It's the number 5 carbon which connects to, this is a phosphate group. And the number 3 carbon attaches to the next phosphorus below it. So it looks like this. It kind of runs in an angle. There's your 5 carbon. This is the nucleotide. But if you notice, before you hook them together, what is H2O?
Sure.
Do you remember how you move water from compounds and make them hook together? Remember we talked about digestion? And you got to put the water back? And because the plant moves the water to make it come together? Well, if this is by itself, and this is by itself, and if you look carefully, I wish I had done it. You see right along there, the H, HO, H, HO. All of this to right here is one base. And this is another try. Now, to make these two together, I want this O to become available to hook on this thing here. So I remove this water. I remove that water right there. I remove this OH from one nucleotide. I remove the oxygen, the H other one. Well, I take out the water, and this line can hook to that 3 right there. And they connect like this. And right here is where they're connected. That's what's together. But still, the number 3 phosphorus, this P belongs to him. Now notice, is this P hooked to that carbon or to an O? This P.
To know.
This is your go-between. This is your natural commodity right here. And to make that thing separate, what do you put back in here? You put back the water you took out. You put back the water, then this H of the water will combine with him, he'll fall off, and that line open, the other half of water OH hooks on there, and you're back to these 2 separate things again. And if you look how it builds from 5 to 3. Now, I'm not showing you, on this one I showed you a lot more stuff. I'm showing you what you need to know on this one. A would be adenine. Okay? This is the nucleotide. I got a phosphate group, I got a sugar, and I got a base. And when they come together, here's the first one. Now, what's going to happen here to make this one hook on? There's H right there coming off. There's H right there. That I'll show you. But this H and this OH will go away. This O is then free to hook on to the 4th one. He becomes a 5. So as you build these things, you build them this way. You build them from 5 to 3, like I said. You go backwards to the side. So if you're going to take this and build it, do you build it in this direction or do you build it in this direction? And look, you know, let me see that. 5
3.
to 3. He's my first member. He's my second tack, my second tack, my third tack. Now if you look at this carefully, was it built this way or was it built that way, this side right here? Did he go, is he the first one ever Or is he the first one ever? You think he is? And you're right. Is that right?
No, 5
You're
and
wrong.
I'm wrong.
You're wrong.
Supposed to go from there.
Notice how the line points this way? He's the first one. 5
Right.
builds to 3. If you look at this, he would have to be the first one. He'd be the second one. And I'm building 5 to 3, 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 5 to 3. So now, if you're going to build this side, do you build up or down?
Go from the up, down.
I got to build this way.
Yeah.
So when these things separate, and I'm going to replace the bases, these bases are hooked on, like, a G goes here, right? But this G comes in first, that T comes in next, that C comes in next. And now when you build this side, you go up. You always start from the 5 side, and you build to the 3 side. So you don't just put them on there just like you lay tile on the floor. So you always start with the 5 in and build down to the 3. So actually, you build to the flat side. You build to the flat side. And that's when you build into the flat side. So this one would not be stuck on up here. It would be stuck on down there. Well, I just said that. I'm going to move her over there. I'm talking now. So you get longer from the 3 in. That's why he would be attached here. He'd be number 5. Okay? Problem is, though, these things are wrapped like this in your body. They must be unwrapped for them to actually do that. And this is an enzyme that makes it unwrap. It just twists it. Now, how you twist tiles on a loaf of bread and you untwist them? Well, that's what you're doing. You're untwisting the ties, and so that thing lines out. Now, this is how it looks in real life, right here. It looks twisted like a spiral staircase. For this thing to work, it's got to be untwisted flat like a stepladder. Okay? And the topoisomerase is what does the untwisting. And all it does, now remember I told you now, there's your 5 side and there's your 3 side. Now when I separate this, my new members, they come on from this direction or from this direction. Is it A or B? How do they build? They build 5 to 3. Problem is, no problem doing this way, but here you got to skip. That makes it complex to build the other side. And we'll get there in a minute. But this green thing represents that's an enzyme. Now on my key here, I decided to let if this is A, that's got to be T there. And if this one is C, then the light blue has got to be. So wherever I put this color, I had to put this color because these are my colors for C and G. And they're going to unzip like a snaggle tooth when they unzip. But the problem is though, first, got to unwind it. Then you can unzip it. Now, there's an enzyme which, like a wood splitter splits wood, that unzips that sucker. This enzyme is unwinding it. Helicase is unzipping it. Right down the middle. And as you see, they're different sizes. And see what I did? I took this picture and I colored it in.
I have a question.
Yes.
Does the topoisomerase unwind it from 5' to 3'?
Mm-hmm.
And the helicase does?
Actually, this is unzipping both directions.
Uh-huh.
You don't have just one tropo. You got one going this way, one going that way.
Okay.
So it's like a busted zipper.
Okay.
or had 2 zippers in it.
Everything okay.
So it's going both directions. That makes it fast. And as a matter of fact, if you had a long strand of DNA, it's doing it here, doing it there, it's doing it there, it's doing it there. It's not just, it don't wait, it just, well, I'll do some here. So actually, it's happening 4 or 5 times. It makes it faster that way.
Same time, okay.
So if I say we're gonna braid this thread, and you braid this amount of it, and you braid that, it's the same thread.
Mm-hmm.
Well, I think I braid the whole thing that way, do it. I mean, we got 3 of us braiding. Well, we have 3 unzipping it. So it's going both ways, and I got a better picture to show you. But I'm trying to keep it as simple as I can. So,
thank you.
So, well, this is more complex than we've done so far, I believe. Now, remember now, there's your 5. Now, as they unzip, 5 goes 3, right? That's easy. But here requires it like do a little bit and start over, do a little more and start over and do a little more. Makes it more complex. You got a leading in and a lagging in. But I'm ahead of myself. Hang on. All right. So there's your zipper, and there's your straightener. So this is your unzipper. This is your helicase, and the topoisomerase is your unzipper. Okay. Now, you see, I'm doing my wisdom. I didn't get all that one off of it. But I need the 3's down there so you understand that from my picture over here, they build from 5 to 3. So 5 is the oldest one. 3 is the newest one. So if you look at this one right here, I'm going to build from 5 to 3, but actually the 3's over here. The 3's over here. The 3 on this side is going to be opposite of 5. Have I lost you or not? All right. See right here how this is 3? 3 versus 5? 3 versus, what would that be right there? 5. Now, this is polymerase. This is going to glue the missing bases. He moves this way. As he moves, he's attaching bases. So as fast as he unzips, he just zips them back on there. Because he is doing it 5 to 3. See, the new ones are 5 to 3. Not the old ones, the new ones. 5, next one, next one, next one. He's going to keep on going until finally it catches back up. This side can't do it that way. Okay, but right now, and see how it's rewinding? So this one is going to look just like that one. And so is that one over when it gets done. When I get done, I will have 2 of these suckers working behind me from that one right there. I will have made 2 DNAs. And I took it off so I could put writing in there. Okay? So this is your polymerase. This is the one that's actually hooking it back on. So when you see an A, what's he put on top of it?
T.
T. When he sees the G, what's he hook onto it?
C.
C. And he goes from 5 to 3. This is going to be a 3. He can't go this way. They can only go that way. So they got to keep starting over. And we'll get there in a minute. This is the east side here. Because as he ends it, he just, it's like, you know how they pave a road and they all go in the same direction? That's how that is. Now, helicase, I made some major work here on this one. It took me a long time because I had to make this. I'm going to take helicase and split this into this. So that I got one side, and notice that's 5 to 3. You point to the 5 every time, point to 5. And when I click it, these are the missing units. Now, I'm going to build it this way, right? Because I built it based on these. So what I'm going to do now Which one's got to go right here? A what? All right. Which one must go right there? What goes opposite of A?
T.
Notice I'm always building 5, 3, 5, 3. 3 is always the end, isn't it? Okay. Which one goes right here?
G.
I see a G. I see a C.
Is it G?
So I gotta take a T over there. And they're building up the page, aren't they? Always, the last one in, 3 3's on the top, isn't it?
Yeah.
Now what's gonna hook on to that one? That.
T.
I got more T's left?
Yeah.
Alright. What's gonna hook right here?
C.
Notice I'm always going, the three is always flipped over every time.
And the A.
Notice how, see a 3 right there?
Mm-hmm.
What's right there?
5.
Well, if this is a 3, what's that right there? 5. I'm always building onto my old 3. Now, what's going to go right here?
The first one, A.
And notice I'm building always. And I've got one more to go. Now, when I get done, I've just separated, because you know, see, I fixed this one. I fixed this one. So now he is fixed. If I start to show again, you'll see that this is what I've just added. And that if you look carefully, this one, not this one has just made himself whole, right there. Now, the other one is the other side now, remember? Because I fixed this side, didn't I? I got to fix that side too, don't I? Yeah. If you look at this one over here now, let's stop the show again so I can make it work. How am I going to build it? Where's the first one go first? Top or bottom? I got to build
From bottom up.
5 down. Well, I need a 5 over here, don't I? But this goes right across. Remember? I build 5 to 3. That'd be a 5 here. But which one ties to the A?
T.
I'll take this one. Notice I'm building, see that 3 right, see that 3 right here? Hooking to that one, I'm building 3 to 5. How about this one? What's going to get him? The G?
Yeah.
Notice I'm still going 3 to 5. So I'm going 3 to 5 every time. What's going to go this one? I'm still building. See that 3 right there? I'm building 3 to 5. I'm building 3 to 5. 3 to 5.
C.
And what goes here?
Let's see.
Now I'm still building it. I'm hooking a 3. I'm sliding a 5 into a 3. I'm building 3 to 5. 3 to 5. What goes here? What goes on that one?
It's A.
It doesn't matter which direction you go. And I'm building 3 to a new 5. 03 to a new 5. What goes here? obviously, I built it that direction. Now, when I get done with it, these came in, and now you'll see that I have produced myself one just like I had. So when I get done, I've got it. Now, on this one, which one was the old half and which is the new half? Is A the old half or B the old half?
B.
A or B?
B.
And I built it in what direction? 5
to 3.
to 3. And what I'm talking about, I brought in this 5, hooked to that 3. This 5 hooked to that 3. So when you look at it, if this is the new member, who was the last one to enter the party? The one on top or one on bottom? Huh? Fitting. Huh?
We're on the bottom.
So I brought a new one in where we had to go, top or bottom. If I brought one more in, where'd you got to go, top or bottom? If I found one more to tack in there, would I tack it in to this one, or I bring it into that one? I got to bring it into the 3 because the 3 hooks to the 5. 5 will not hook to a 3. Don't know why. It just doesn't do that. I went looking one time and said, well, what's the difference? Well, here's which way it goes. But it does in biology and matter. I don't know yet why it matters. I just know that the order is from 3 to 5. And what's confusing, you want to think this way. You want to think 3 to 5 like this, don't you? Can't do that. This 3 to this 5. This 3 to this 5. This 3 to this 5. See, that's a 3. What's that one? I'm still doing 3 to 5. I don't have a Bobby Temp rule for that yet. I'm getting close to one where I think I build them with a point sticking up every time. If a rule becomes, I want to put the point up every time. The point under the one above it, right? If you put the point under the one above it, would that not be the Bobby Temp rule? Because if I go back in time, well, he was over there with me. All right. The Bobby Temp rule would tell me I want to poke the one above me. Right? If we're going to poke the one above me, then I got to put it, if I had nowhere to go, if I want to poke the one above me, I got to start here so I can poke him first, can't I? If I start here, not that way. If I put him first, can I poke him by putting one above him? I got to poke the old one. So my mind saying, I got to poke, okay, I got to put him first so I can poke him with the next one. See that right there, the arrow? And if I'm going to poke him, I got to put this one so I can poke him with this one. And Bobby Temp rule is stupid, but it's working. Because I am poking the one I put in there last. Right? So the new one always pokes the old one. And if the new one always pokes the old one, this one will poke him. If I go back to the one I did earlier, will my Bobby Temp rule work for that one? Now, if I'm going to build these suckers, and my rule says poke the one that's on there. If I want to poke the one that's on there, which one had a button put on first so I can poke him? The one on top or one on bottom? If I put him on first, can I poke him with a new one? I can't go on top of him, can I? So I must put this one on first so I can poke him with the next one. And I can poke this one with the next one. And I'm still doing 3 to 5. You see that 3 right there? 3 to 5. 3 to 5. And if I ever get my mind, if I ever get thinking too hard, I get confused, I'm going back to my poking route. Gotta poke the old one. If I, you know, if I do this number here, and I put him right there, and I say, put on the next one. Which one you gonna put on? One on top or one on bottom? What's my rule say?
Poke the old one.
I gotta poke the one on there. If I'm gonna poke him with the arrow, which one goes on? The one above or one below?
The one above?
And I gotta use this one to poke him. Because that does give me my 3 to 5 direction. See my direction? Now if you don't, if you get confused about it, I see all these 5s. I see a lot of 5s. Which one are you talking about, Bobby? I'm talking about the one right here. I want this 3 to go to this 5. And the poking rule, I want this point to poke the one already on there. If I put him on, he ain't poking anybody, is he? I want this one to poke that one. And that's still my 3 going to 5. And I want to poke this one, so I gotta use that one to poke that one. Stupid, but it'll work. If I get confused on my numbers, I'm going to my poking rule. And I'm telling myself, the one I put on new is I got to poke the one already there. If I remember that, oh, no problem. To even start it, I got to poke the one I put on. So I put this one on first. I can't poke him, can I? So I got to put this one on first, so I can poke him on the next one, and then I'll go right on up and I go. And I do it. I'm still going, and notice I'm still doing 3 to 5. I'm not using these ones. I'm not using these. I'm using the ones I put in up. 3 to 5. All right. That makes sense. That's confusing, I know. And I'm going to go figure it out. The Bobby said poking rule. But I need that myself to help me, because I start looking, my eyes go across, I go, oh man, I see a ton of 5s, I see a ton of 3s. He said hook a 5 to 3, they're all over the place. Poke them. Poke the old one with the new one, and you'll get it right every time. Okay? So, we just got doing all this stuff here, and now we're on this one. Now, this strand is replicating, and this strand is replicating.
Mr. Tuff, do you want to stop here?
25, 26 after.
Oh, I'm sorry. I don't know what I'm doing. I do want to stop there, because we've done enough right now. We'll come in. I'll go home and put on this right now on the angel. I'll do it here by leave here, because I got here. Then I'll go and finish up your lab. I'll put lab on either late tonight or towards late tonight, because I'm almost done. So be watching for it. Come on. But it's going to be, I think it's very simple. It's got a microscope, I got slides, and got a worksheet you're going to work through like I like to do and make you think. So be watching angel. But I would tell you it's there, so you got to keep looking for it. You got to search. When the lab's on there, I will indicate it. So y'all take off.